All right. Hello, everyone, and welcome back to another edition about the Truth About Your Health podcast, where we teach exhausted and burnt out adults the truth about their health so that they can get their energy back quickly. And what a joy it is to have Martha Carlin here today, who is the founder, and I love the term, the chief executive revolutionary for the BioCollective company, where it aims to accelerate microbiome research by supporting robust, reproducible, and reliable results that allow the field to realize its full potential of life-saving discoveries. And she has a very unique story where in 2002, her husband, John, was diagnosed with Parkinson's disease. And well, I'll let her tell the story from there. So Martha, thank you so much for being here today.
Thanks for having me, Joel.
Yeah, so I'm excited because you and I say this in an enduring way, I think are both nerds, and nerds found each other at the Biohacking Congress, and we started talking, and we probably could have talked to hours and hours. And I think we had a couple of the times, maybe the presenters look back at us during their talk and it's like, hey, keep it down back there in the back row. So here we are today, and I really want to hear and have you share with our listeners just the genesis of where you got to today, and maybe sort of a shortened version so that we can spend more time explaining what you've discovered along the way. So why don't you just tell us your journey, Martha?
Sure. Well, my typical background is in business operations. I was a turnaround expert and a head of operations for a large real estate investments trust. So totally out of the science field. In 2002, my husband, John, at the age of 44, was diagnosed with Parkinson's. And, that's pretty shocking when you're young. We had two small children, and I had an older child. And, I, being a turnaround expert, I sort of looked at how the doctors, it was kind of like they just drop a bomb on you and say, here's a pill. We'll see you in 6 months. There's nothing you can do. And that's just not who I am. I looked at it, I said, this is a systems problem. I need to understand the system in the same way I do a business. So I've got to start teaching myself science. So I started, and I really looked at food first, because that's, food and water are the major flows through the body that really get ignored by traditional medicine. So I spent about 10 years really studying food, nutrition, how we grow our food, the chemicals that are used on that. And then along the way, came up to 2014 and read Dr. Martin Blazer's book, Missing Microbes, about the rise of chronic disease post antibiotics and how the antibiotics were destroying the microbiome. And a few months later, the first paper was published showing that you could map the two symptom phenotypes in Parkinson's to the microbiome. And I was like, eureka, that's it. I quit my job. We sold our house, started funding research at the University of Chicago, looking at time series. And part of my interest in the type of sequencing we were doing, which very few people at the time were doing, whole genome sequencing. The microbiome has about 300 times more genes than the human genome. And I felt that that was going to be the map to tying out some of the problems from genetically engineered food, to be able to see that, all the genes in the map. And so, we've done over 150 whole genome samples of John's microbiome. And I went from there to founding the BioCollective with a researcher from the University of Chicago and a virologist, Dr. Suzanne Vernon, who had spent 17 years at the CDC studying chronic fatigue and viruses, so that we could build a biobank of fecal samples to connect the dots across disease, not just in Parkinson's. But along the way, I actually have become I think a pretty good scientist. You call me a nerd. I'm a super nerd.
Yeah, I said it with love. So to break it down to, just for my brain to understand this, I love what you said in terms of food and water, and taking it from a layperson at that time, where you're certainly not now, where, okay, my husband has this, I got to break down the system and look at the food and the water and how that is I guess assimilated and where it meets the inside of our system, which is our microbiome. Can you explain what you meant in terms of mapping the biome and seeing when you had that eureka moment? So repeat that again in terms of an easier way for the person that may be lay and they want to understand. Okay, so what did you do? You found that there was a model that shows that these are the Parkinson patients type biome, and this is what a normal biome is, and then compare the two. Is that pretty much like how did it go wrong, or what was the difference between the two?
So, the initial study by Dr. Philip Schepperhans in Finland they take fecal samples and they sequence those and compare them to a healthy microbiome. And what he was able to show is, there's a difference in a Parkinson's microbiome, and then within Parkinson's, they can be subdivided to the people who have tremor dominant symptoms versus people who have posture and gait. And from that point, there have been hundreds of microbiome papers in Parkinson's since that time, advancing, looking at proteins, looking at, other mechanisms related to, nutritional deficits and, how the microbiome produces neurochemistry. So, it's just been a crazy emerging field since 2017.
Right. And then the next thing that you mentioned in terms of how much orders of magnitude the DNA for the microbiome and bacteria is compared to the human DNA. And it's, like you said, an emerging field, and it's almost overwhelming to be aware of what you don't know, right? It's like, oh my gosh, like as soon as you've had that eureka moment and now you find out all these other rabbit holes, and you're not just comparing a Parkinson's patient to a quote unquote normal sample, but Parkinson patients that have this versus that. And then there's so many different other rabbit holes to go down. And so I would love to discuss with you what you're learning sort of from there, which is a slippery slope because there's so many ways we can go. But just to recap, you said, okay, I have this eureka moment. And so I'm going to do like the turnaround person that I am. I'm going to go all in because it's my husband and his health and life is on the line. And I want to make sure that we figure this out. So you started the BioCollective, and you have different testing procedures and different ways to figure things out. So kind of go from there. Like, tell us what was the next, how things unfolded and where we are.
Sure. So we started out, it was just really early in the microbiome space, but we collected whole fecal samples. So, we designed a kit and patented it and started collecting a biobank, if you will. And, one of the more interesting things we learned in the process, our lab people could actually tell a person with Parkinson's from looking at their fecal sample without knowing they had Parkinson's. It was that distinct from other samples coming through the lab. And the key tell was a lack of water homeostasis. So the fecal samples were more like concrete, and a diagnosis of chronic constipation can precede a diagnosis of Parkinson's by 10 to 15 years. So I think, and there's nothing in the published literature about this water issue. So that was a really interesting learning that sort of provided some clues for us of where to look. But, we were doing the genomic sequencing and providing almost a 50-page report of all these genes and information, and people would take it and try to have their doctor look at it. And it's really too much. So after we built our biobank to a point where we had a sufficient amount of samples and data to start to connect the dots kind of across diseases, but we had 100 Parkinson's patients. we stopped collecting samples and then really started digging deeper into the analytics of what is going on in the microbiome, what can we see, indicators. And from that, one of the more interesting things, we did some machine learning with a Polish company called Artigen, where we found these biomarkers, and one of the key markers was in glycosylation, which, for people, if they're not familiar with that, it's basically the decorating of proteins with sugars. And, one of those is hemoglobin. So it ties back to hemoglobin A1C. And, from that, we went back into the literature and saw this connection in the Parkinson's research of comorbidities in glucose metabolism. And there's, I think, about 65% of people with Parkinson's have some insulin dysregulation component. And, there's even an area of research called looking at what's called type 3 diabetes, which may be insulin resistance in the brain.
Yeah, I mean, again, I'm loving the genesis and the growth of it, and I'm following the, as you tell me. Okay, so we started to produce a collecting method, and as these samples came in, we were able to know, without even comparing them to the norm, that this person had a Parkinson presentation, because there's so much similarity to what the Parkinson, I guess, the characteristics of that sample look like, to the point where we finally had enough. And now let's actually do some studies and figure this out. And this is where we are now, and looking at the inborn error of energy production, right? Which we look at as cancer, as a metabolic disease, and ultimately any neurological disease or any health challenge is a metabolic breakdown. And ultimately, your cells are not converting the food you eat with the air you breathe into water and energy. And on top of that, like you just said, the foods are adulterated. There's not a lot of minerals in there. And tragically, there's so many chemical disruptors and things that you were just talking about. And then your body isn't able to use oxygen effectively to produce energy. And then it has a way of making energy more efficiently by fermenting glucose. And then that has an imprint on the biome. So let's talk about where you want to go from there, Martha. There's so many ways we can go. Yeah.
Well, one of my early focuses, going all the way back to the beginning, was looking at the herbicide glyphosate and how glyphosate is used in farming. And there, it translates out in a number of ways, because the bacteria in our gut are producing a lot of those vitamins, hormones, and neurotransmitters. And glyphosate actually selects for a more pathogenic profile in the microbiome and destroys some of those key vitamin producers. And so, that was an area that I was, really interested in, is trying to sort of figure that out, the glyphosate connection. The other thing, and I know you talked about this on some of your other shows, is, glyphosate was originally patented as a chemical to clean scale from pipes. It's a very powerful chelator. And so it's not only chelating metals from our body, it's chelating metals from the soil, it's chelating metals out of plants. So, we're just getting less and less nutrition. And I discovered a book by a researcher from the Cleveland Clinic, Dr. I think it's Derek Lonsdale. He's no longer alive, but he has a book called, I think it's called High Calorie Malnutrition and Thiamine Deficiency. So again, back to those B vitamins, which are made by the gut bacteria. you have this interconnection of what's going on in the food supply and how that's impacting the microbiome. Also, in in that same pathway in the bacteria, the first enzyme in something called the shikimate pathway is a copper dependent enzyme. And, copper is depleted because of glyphosate. And then further downstream of that biochemistry is dopamine production. So you're impacting, dopamine production as well through those foods. And so along the way, just trying to figure out, okay, how do I maybe solve some of these problems? I actually attended a conference where researchers from Israel were showing that the sugar alcohol mannitol could stop the aggregation of the proteins that are the hallmark in Parkinson's. And in this animal model, they could actually pull them out of the brain. Well, mannitol is a really interesting molecule. It's a very powerful free radical scavenger. It's used to treat brain swelling. It's used in like the delivery to enhance the delivery of medication across the blood-brain barrier. It's also used in scientific research to keep the mitochondria happy. So, I came back and actually started studying mannitol chemistry and found, oh, wow, there's a small handful of bacteria that produce mannitol from glucose and fructose. I wonder if we could put a factory back into the gut to make mannitol. And so, we prototyped a product and it actually converts glucose and fructose into mannitol, which humans don't use. We eliminate it. And so that was sort of my first step in trying to figure out how can I do something to help John. And at the time that we made that formula to test for him, he was walking with a cane and not doing as well as he had been. And within a month, we're measuring his microbiome and we could see it moving back closer and closer to the healthy human microbiome profile. And in about 30 days, he was no longer walking with a cane. So, we took that concept of, how can we start to look at the microbiome as something that can be supported and modulated. And in our formula, we actually had a focus on identifying strains of bacteria that were good vitamin producers, but could also withstand glyphosate because you can't get away from it in the food supply, and break glyphosate all the way down, because in the biochemistry of glyphosate, when it breaks down, most bacteria break it down to something called AMPA, which is toxic to the brain, even more toxic than glyphosate. Whereas we have a strain of bacteria that breaks it all the way down to carbon phosphate and water. And so, you're back down to your elemental level where you can eliminate something without harm.
No, it's amazing information. So the way that I think about the next question is that, okay, how if we have something now, again, just sort of a quick response from you, what would be the purpose why would they even put glyphosate in our soils? Was that because they want to make sure that they're killing pests and things so that they get better crops? Is that what was the purpose? I mean, from just an outer laying sort of feedback?
Well, glyphosate is a very powerful herbicide. So, for simplification, they can spray it on the fields, and they engineered glyphosate resistance into corn and soy and some of these other crops, so that they could just spray the whole field. If you think about how crops have been grown and people have to spend time weeding and doing things like that, they were trying to make it more efficient. So by engineering resistance genes into the plants, then they can spray the field, and only those plants that are resistant, the corn and the soy would, survive. and the weeds would die. But what we've seen over the last, 30, 40 years is the weeds have developed resistance as well. So then you get, more use,
Right.
more chemicals, more, and, glyphosate isn't the only herbicide or chemical that's used on the food. And, there are many mechanisms. If you go back to these herbicides that were actually nerve gas from World War I and World War II, that, the chemical companies looked for another use and put them into agriculture. But, if you look at how those nerve gases work, they're messing with acetylcholine receptors or, other neurotransmitter mechanisms in the body. And now we're putting them on our food and eating them all the time.
Yeah, I mean, it's helpful for you. Thank you for sharing the beginning of what the thought process was. To ultimately comes down to the dollar and how could we get better yield. But the ignorance of, okay, well, it's going to be resistant, so nothing else is going to be problematic with that. Ultimately, when we eat it, then we have the same engineering in our microbiome that's been designed to impact the crops and make them resistant to glyphosate, but yet at the same time expose them to glyphosate and mess up the diversity in our microbiome to not be able to break down the glyphosate or impact the dopamine and different areas in our body that ultimately helps us make energy at the level that we need to, and then becomes neurotoxic and a whole slew of other downstream impacts. And then from your research, you saw that mannitol is one of these things that can help break down glyphosate more effectively. And how can we, or not so much more effectively, deal with it better or be able to?
Yeah, well, the mannitol is actually not what's breaking it down. It's actually, so we have a species of Lactobacillus plantarum that has specific genetic function that enables it to break the glyphosate all the way down.
Right. So, and good, I appreciate your scientific mind because, well, that's not actually true, Joel. It's this that does that, but the glyphosate helps this in that area. So that, right. I mean, the mannitol that helps. Yeah.
The mannitol is a free radical scavenger. It's a 6-carbon sugar and it's picking up hydrogens, but it also modulates pH. And if you look in, the literature on fermentation production of proteins, you actually, the more acidic the pH is, the more protein production you get in a fermentation. So the mannitol is a neutral molecule and it's modulating pH, which is probably part of that mechanism. Altering pH is altering the proteins in the gut.
Right. And then I'm sure as we continue to talk, there's other important ingredients in formulations to get and continue to grow, to be able to address as many physiological impacts or breakdowns or improvements as possible. I just had an aside, and I know there's the Biome company that does the whole sequencing. What's your feedback on that? What's your sense on what they do?
So, I think there's Biome, there's a company called Day 2 that was, based on some research between the Weizmann Institute and the Mayo Clinic. They're taking different approaches. I mean, it was interesting to me because I sent samples to both Day 2 and Biome from the same fecal sample and got very different food recommendations. Now that was 4 years ago, and I think that the field has continued to advance, and certainly Biome has they have a lot of funding and they're doing a lot of research. So I think it's an interesting concept. I do think that we're still a number of years away from being able to, perfectly and ideally map a solution for somebody from looking at, certainly not a single microbiome sample, because, the gut changes based on your diet and, from week to week. I mean, you can alter your microbiome in less than a week by shifting your diet to a high fiber diet, or, going to a ketogenic diet. You're going to change your microbiome dramatically.
Right. And again, I appreciate it. I always say I do these interviews for myself for information, and then everyone else that's listening to it gets to hear me over my shoulder. And I'm like, oh, I'm taking notes. So as far as the concept with the Biome company is, hey, we'll do a sequencing of your current real-time microbiome, which would be different from days and weeks and months and years ago, based on what you ate, what your stress levels were, where you traveled to, all of the above. And there's so many dynamics. But what's the thought process, Martha, in terms of how are they extrapolating that sequence and saying, okay, these are the foods that you'd be better to eat versus not eating?
Well, Biomes it's proprietary. They do something called transcriptomics. So, theirs is quite a bit different than somebody who's looking at genomics. So,
right.
I think it would probably be better if you could maybe get somebody from Biome to
right. I'm just hearing
than for me
to
really speculate about what they do. I mean, it is interesting because I think actually it may tie in a little bit to some of the work I've done in Parkinson's in something called molecular mimicry, because they are looking at RNA viruses, and there were some things that, like, they told me not to eat green beans because there was a specific mosaic virus from the green bean that might be a problem for me. And I actually got interested in molecular mimicry from research of a Dr. Robert Friedland at the University of Louisville, who showed, so people who smoke don't get Parkinson's at the same rate. And I'm not recommending that anybody go out and smoke. But what Dr. Friedland found was there is a tobacco mosaic virus that mimics something that is connected with Parkinson's disease in a way that, if you've come in contact and you're carrying that tobacco mosaic virus, it's almost like a vaccine against Parkinson's. So that was a really interesting area as we started looking at different microorganisms in the Parkinson's gut and sort of mapping that into this molecular mimicry concept. Bacteria produce something called a heat shock protein. I don't want to get too nerdy on you, but it's a little peptide sequence that it's kind of a danger signal. If the bacteria are under stress, they're putting out this little peptide, and those sequences, it's a series of amino acids basically, or the body then recognizes that, creates, the immune system reacts, and you get antibodies to this heat shock protein. Well, as it turns out, there's a number of those that are similar to human proteins. And so you can get this autoimmune reaction. And the same thing is, there's also similar heat shock proteins in plants. So you can sort of get this perfect storm. And one in particular that has been of interest to us is something called alpha crystalline, which it turns out that wheat has the most closely aligned alpha crystalline to human alpha B crystalline, and mycobacteria species also produce that. And they produce another heat shock protein, or they produce another peptide sequence for something called cardiolipin. And so we started looking at those pieces in this concept of molecular mimicry and what might be going on in the immune system with people with Parkinson's having kind of what I characterize as a chronic low-grade infection of multiple organisms, bacteria, fungi, viruses. And all of that goes back again to an unhealthy immune system, because our nutrition, we don't have the minerals, we don't have, we don't have the vitamin production. The body is just not well tuned to fight off these pathogens.
Yeah, amazing. Thanks for bringing it back to that. And that's what we talked about earlier is that if you don't, I really do believe we have the inborn innate power to heal ourselves when we have the right minerals and nutrients that is grown from our earth and not from a lab and patented and made millions of dollars from. But then on top of that, the downstream effect of chelating minerals from herbicides and pesticides not only strip the foods we eat of these healthy, vital ingredients, but it also messes up our factory and our ability to make the minerals from our microbiome. And then ultimately we have stress responses or beacons sent out by our bacteria. And we ultimately create a response to that, which is perhaps an immune response, which is an antibody, just as if I had an exposure to a virus, my body creates a antibody to it. Or if just the way that vaccines works is that concept. If you give a small little attenuated dose or whatever new ways they're doing it, you get an antibody. So you've developed an immune response, which isn't your inheritable immune response. It's an acquired immune response, which now you have an antibody to. And the mimicry is very similar. If the protein structure in wheat is very similar to the protein structure in your gut or in your brain or in your skin or wherever they may be, and you've developed an antibody to that peptide or the protein structure in the food source because of a stress response, because of adulteration of our food supply and lack of minerals, now you're creating an attack against your own tissues, and that can cause fill in the blank autoimmunity. So I guess from there, take us wherever you want to go, Martha, on that.
Well, I think if we go back again, you hear a lot of discussion, in the last, say, 4 to 5 years about something called leaky gut. And part of that mechanism of activating the immune system is when these are crossing the gut lining because it's, quote, leaky. Well, if we go back and look at the bacteria that that we find more prevalent in the Parkinson's gut, there are actually a number of organisms there that produce something called pore forming toxins or different kinds of toxins. And what those toxins can do is, form little holes in your cells. They can affect your gut lining. And so, that's just kind of another piece of that puzzle that is exacerbating that, vicious cycle loop from the gut, the immune system, the overactivation, and the, dysregulation of everything in the microbiome. And, we tell, I say, I knew instantly, when I read that Parkinson's paper and the book on the antibiotics that, that was just such a eureka for me, that my training is as an accountant, and, the gut is the general ledger. I mean, it really is that record, and we can start to see, especially, when we have this very detailed genetic information, you can start to really, map these pieces of the puzzle, back into the contributing factors to what's going on, not just with Parkinson's. I mean, it's really evident across diseases in our data. And we've looked at that. We've looked at IBS, we've looked at smokers, we've looked at cancer. So, I think what we're going to see as this field continues to emerge is, an incredible acceleration of understanding how these things are impacting the gut bacteria and how important. I mean, really, we're more microbial than we are human in terms of how we function. And if we don't have that functional diversity, we can't be healthy.
Yeah, I love it. I have a quote down: "The gut is the general ledger." That's an awesome quote. And as far as a couple of things that I think about is, again, as complex as this is, the solution is to get nutrients and minerals that we've been made to have, and as best we can get them from what's grown in our healthy soils. And you don't have to be a scientist to just have really nutritionally dense foods that allow our body to recognize that and not create stress alerts and not create antibodies and have cross reactions and create autoimmunities, but actually get the food from our sources and let the body heal. That's one thing I think about for people that are listening. Sounds, you have something you want to add to that?
Well, I mean, so I really, when I started seeing what was going on here and tying it back to my original digging into nutrition and food and how our food is grown, had a clear understanding that it's going to be very difficult for us as a population to fix our guts if we don't also fix our food. And I'm very fortunate. My chief scientific officer of my company is a real gem. Dr. Raul Cano was a professor of microbiology at Cal Poly for over 30 years. And he was a pioneer in using basically microbial ecosystems, putting microbes together to clean up oil spills. And so we have another company outside of the BioCollective called Paleobiotica that has taken a collection of his ancient microbes that he revived out of amber occlusions and deep sea cores, and has combined those into agricultural products that we're bringing to the market. This year we'll do a small scale test, but we have filed a patent for the bioremediation of glyphosate in soil and water using our microbial systems, and for improving nutritional uptake in plants, plant growth, all of that, because we've destroyed the biome of the soil, and all of that is, it's a chain reaction. So we must restore the soil to restore the plant nutrition, to restore the animal nutrition, to support our nutrition. And you can start growing some food in your backyard, and we're doing a little sample on our BioTiquest website where the product's called Yield and Shield. So in your backyard garden, if you are interested in trying a microbial solution to help support your soil health, we're going to have that launched here probably in another week.
No, that's awesome. I love the rabbit holes and just how you've gotten bitten off more than you can chew when you start to realize, oh my gosh, now it's the implications on saving the earth. And it really is, from the point of view, you can't just, you could try to grow your own food, but as a society and as a world being, you have to restore your earth to be able to get those soils back together and heal. So I love that, Martha. As far as I don't know if you stuck around for the whole conference, but Dr. Pompa had talked about how when we had the oil spill in the oceans, and we had Chernobyl, and we thought our earth was, and probably even what was it in Japan,
Fukushima.
Hiroshima, we feel that will impact the earth to the point of no return. But then we have this inborn ability of hormetic stress, right? Where our body gets a little exposed to something, but it makes it stronger. And we found that it ultimately did. So I guess the question is, how much does hormetic stress, even though the fundamentals need to be replaced, does that impact our ability to deal with this so that it's not the sky's falling and we're forever past that point of no return?
Sure. Well, if you look at something like whether it's Chernobyl or Fukushima, it's a aggressive assault, and then a stepping back from that. And so the ecosystem rebounds, through a selection, and, microbes evolve much faster than we do. So they're able to, evolve and figure out what needs to be done and help restore the ecosystem better when we just leave them alone. Where,
Right.
Where our biggest issue we have, I think, is still this, on our food supply side, it's still this sort of, these chemicals we're using are not a problem. And so it's not hormetic stress, it is constant stress. So, the hormetic stress is like, I'm gonna like push you, or I'm gonna fast for 48 hours, or, something like that, as opposed to I'm constantly assaulting my system, with something that's toxic. So I do think we have to be, very thoughtful in what we put in our body. We are made of the molecules we eat. And so, when you're about to put something in your mouth, you should think, do I want to be made of this? And, one of the things I talk to people a lot about is sugar, because, glucose and fructose are, such damaging molecules in the way that we're ingesting them. And I know you talked to Morley Robbins about how fructose blocks copper uptake. But, one of our key markers in Parkinson, again, is, this glycosylation. And, I know many people with Parkinson's who have, sweet tooth, sugar cravings, they're carb loaders. My husband was a marathon runner, so he was trained to be a carbohydrate engine in order to run those marathons. And, I think just really starting to think about this low carb living, and what sugars are doing to us. And that sort of brings me back around to that, the mannitol product that we made for John. I mean, one of the key things that we found from it, I made it to make mannitol, but it actually alters glucose metabolism in such a way that we did a small study where people dropped fasting blood glucose by 10 milligrams per deciliter over about 8 weeks. And we actually have that in a diabetes clinical trial, because I think, sugar feeds cancer. Sugar is a problem in Parkinson's and neurological diseases. Glycation is an indication of, aging. So all of these sort of pieces come back to, what we're putting in our body, and sugar, certainly carbohydrates being one of those things that we need to be more mindful about.
Yeah, no, Martha, that's an awesome answer, especially as far as the hormetic stressor goes. It's not meant to be a constant stress. It's meant to be a short exposure to be able to stimulate that homeostatic allostasis load in the body. And I love the other answer in terms of microbes and bacteria are basically designed to do that. Their intelligence is to mimic what's being exposed to them and account or accommodate for that. So that's an awesome answer. And then I love how you're going down the idea of, well, how can we harness that? Like, notwithstanding everything being equal, like that doesn't change the onus on us understanding what we're eating and making sure that we work to change our soils, and really, I think even deeper purposes have legislative changes as well, which is a Samson or David versus Goliath type thing, right? But as far as, so maybe I want to talk a little bit about the sugar, because I think that's a really important point. And as far as what is it that's inherent in the product, and not just, is it just the mannitol in the probiotic, or are there engineered other, you were mentioning the lactobacillus, like what is in that product that is doing direct impact on glucose?
Sure. So there are 3 primary organisms in the formula that do the conversion of glucose and fructose into mannitol. And that is a strain of bacteria that you don't find in a lot of probiotics called Leuconostoc mesenteroides. That actually, that strain
say that 3 times fast. Let's hear you say it right now.
Yeah. Leuconostoc mesenteroides. It's found in a lot of fermented foods. We isolated it from sauerkraut, from cabbage I grew in my own garden, and fermented and stored for over 4 years before we isolated that organism. It has Lactobacillus reuteri, which is also an oxytocin producer, and reuteri also makes mannitol. And then a strain of bacteria called Bacillus subtilis, which is a spore-forming organism that also makes mannitol. But these organisms, so we have a computational model called BioFlux, where we can predict how these organisms work together. And what we see is we designed the formula to make mannitol, but it also increases the production of butyrate, which feeds the lining of the gut and supports that. It's also altering something called siderophores, which is basically sequestering iron from pathogens and eliminating that from the body. The plantarum, the strain of plantarum actually came from fermented elderberries from my neighborhood. So we've really tried to look for and select strains with genetics, natural genetics. So none of our organisms are engineered, that, have those functions that we see that we need. And then we put them together and run them through our computational model to make sure that as a group, they're going to collectively do what we want them to do. And so, that's kind of, it's an 8-strain formula, but those are some of the key strains that, are powerhouses for us. There's also a Lactobacillus paracasei, and I think that one also helps with the breakdown of proteins, which, could also be helpful in Parkinson's.
Yeah. And listen, I don't think it's just helpful for Parkinson's. 88% of our population is metabolically sick. And ultimately, Parkinson's, even though it attacks certain tissues in the body, it's the same mechanism of being metabolically sick. And it's the same mechanisms of the different strains that impact the physiology favorably that would help anyone. And I'm really excited about that for sure, especially the oxytocin. Just as an aside, when we do genetic test interpretations, one of the main genetic test SNPs that we look at is the oxytocin receptor. And when people have polymorphisms with that, they tend to be more empathetic people. And I always explain that as the first thing that I explain when we go through a genetic test interpretation, because when you have the antenna sticking out of your head a lot more and it's bringing in all these frequencies, it's going to drive that oxygen consumption rate super high. And if you're not respiring at the level you need to, and you're fermenting glucose, and that's creating all the things that we've talked about, given that your microbiome can't do this, that, and the other, it's going to make things a lot worse. On top of that, what's amazing about oxytocin is it suppresses the NADPH oxidase that creates the free radicals, that creates the mast cells, that creates the histamines. I love the idea of fermented foods. Unfortunately, a lot of people that have these major GI challenges, it's a catch-22 because they are already fermenting from a biological point of view. So any other foods that they get that are fermented typically kick that ocean floor. And I know you got to say something here in a second, but at the same time, the fermented foods, if you look at longitarians or blue zone countries, I think they've worked it into their dietary supply to have the microbiome to be able to deal with the fermented foods. And instead of that, could be another general ledger as well, in terms of, if you can tolerate fermented foods, you're on the surplus or you're in the black. And if you can't tolerate it, you're in the red, right? Does that make sense?
It does. The only people that I've had that have had an issue with the sugar shift product is people who are super sensitive to FODMAPs. But, the way our product works, a lot of probiotics are using new capsules and designs to try to get it all the way past the upper GI tract and down into the colon. But I actually, because you're eating the sugar up here, and so many people have SIBO, small intestinal bacterial overgrowth, you want to get those sugars out before they get down to feeding, the SIBO organisms. So, for the most part, people don't have a challenge with the product, even though, mannitol is a sugar alcohol. It's making a small amount of it. But you're doing this conversion and, helping to address, I think, the feeding of the sugars in the upper GI tract. But, what I do tell people, if they've got a high level of fermented food sensitivity, they need to deal with that first by, like addressing the pathogens in their biome. But, one of the interesting things in the data when we did before and after, first on John and then another group of people who took the product, is Streptococcus, going all the way back to some of my observational stuff and talking with people with Parkinson's. People with Parkinson's had quite a few of them had this history in their childhood of having frequent strep infections and taking antibiotics a lot. And so that was sort of interesting to me. I looked at, so I talked to 23andMe, and they also looked in their data, and there was an increased risk of Parkinson's disease with people who had 4 or more strep infections. Dr. Shepperhans went on to show this correlation with antibiotics, and strep is also an interesting organism in minerals and iron in particular. It uses its iron to cross feed some other pathogens. I'm probably going way down the rabbit hole here, but the preferred food for Streptococcus is glucose. It loves to grow on glucose. And what we found in our before and after testing with the sugar shift formula was that Streptococcus was almost disappearing completely. And when we did machine learning with all of our samples across the population, we found that people who had more than 1% relative abundance of streptococcal species in their gut were in some kind of disease category. They weren't all in the same disease category, but the people who were healthy had below 1% streptococcus in their gut. So this alteration, I say, we design a formula that is not just putting in a bacteria, but it's altering the landscape of the gut. And, that's what our focus is, to put a working system in place that can both deliver the metabolites you're looking for, say butyrate, or energy production, but also transform the landscape that makes the ecosystem there, better for a better flora.
Yeah, I mean, that is the true sort, the harnessing the hermetic consequence or the effect of stress and putting that into the microbiome. I almost think of it as calling it a harmonizer, because we have I have a friend that has, he owns Therasage, and they have a harmonizer for EMF. So it doesn't block it. it harmonizes it to change the frequencies so that it's not deleterious to us. That's one thing. But as far as part two, because Martha, we gotta not. I'd love to go forever. We definitely will do part two if you're willing, because I have so many more questions. This is much more rich than I had anticipated. So I know that on top of the correlations, like I think part two would be, okay, now that we've talked about the microbiome, what other correlating epigenetic or genetic factors have you seen? And I think you and I hopefully found something together in that, hey, there's this antibody to parietal cells or this B12 thing that is copper dependent and or needs a lot more, it has a lot more expression. There's also the fact when you and I talked initially, and you were telling me he was a marathoner, and I was thinking about, well, I bet you he wasn't doing it in an aerobic way, meaning he's highly conditioned, but he's also charging his anaerobic energy systems to be plasticized, if you will, and being taught to go down this pathway. So when you feed it with sugar and you put in the glyphosates and you do all these other things, it's that perfect storm. And then, of course, the genetic susceptibilities as well. So so many things we can go with that, and I'll allow you to have a comment here in a second. The other thing I wanted to do or talk to you about was, I mentioned to you, hey, like I also have a lot of clients that have these perfect storm issues of, they also have not the ability to lower glutamates, or they don't have the ability to lower histamines. Is there, in the foreseeable future, putting that all in one product, or would it have to be complementary, Martha, and have like a couple of different products? Like what's your thought process on that?
Yeah, I think we could certainly look at that. Our model is set up in a way so, if glutamine is the target and you want to figure out how to sort of clean that out and get rid of it, we can identify, organisms that can do that, and, look at, can you have a one size fits all, or do you have to kind of take some steps along the way? Right. And one of the other things to really look at is, if there's a connection to what's in the microbiome, then how do you kind of alter the landscape to get rid of the problem that's there to enable, what you're putting in to work better?
Yeah, and I also think you're right too, in terms of, I didn't mean to say like, oh, the people that have an issue with fermented foods shouldn't take the sugar shift product. I think it's more of a tell of constitution. Like, this is, I always say to people, excuse the language, this isn't a friggin' magic wand, right? Like you don't absolve yourself of the responsibility of removing processed foods and sugary foods and making sure that you settle down inflammation and you restore your mineral balance and you get your magnesium levels up and you remove all the things that are draining your all the things that you and I know right. with Morley. So I guess in concluding the number one, because I really do want to do number two, if you will. I always ask my guests, because there's so many things we haven't talked about, what would you like to have known? Because I call it the Truth About Health podcast, because I think there are a lot of lies and deceits and bad, nefarious reasons for doing things, and all profit, in the name of profit. But without that being said, Martha, what do you wish you would have known then that you know now, that you might have implemented in yours or John's health to avoid, or at least maximize the constitution to not be so, I guess, susceptible to something like a Parkinson's?
I think just very early on, I wish I had deeper knowledge about the food and the chemicals that were in the food. I look, my oldest son, who works for me, is in his 30s, and he has a lot of allergies and inflammatory issues. And, I look back at, when he was little, eating Kraft macaroni and cheese and, things that, or, cereal, There's just a lot of things about the food that, I was just ignorant. And so I think that is the one thing, that and the water. So when we started actually looking at the water and filtering the water and, making sure that we're taking minerals, because we're now filtering the minerals out of the water. If I had known what I know now 25 years ago, maybe none of this would have happened. But, hindsight's 2020, and, I'm on this journey. And, sometimes John and I talk about, like, we were brought together, and sometime I'll tell you that story because it's a pretty amazing story. We were brought together on this planet to make a difference. And, I say it with all honesty, I feel that I'm here to save the world. And, I'm doing that through the soil and through the human microbiome and through, all of my mission to educate people and help people to really understand we do have the power to heal ourselves, and we have the power to say no to what's being pushed at us. And, we can take back our land and our lives and our health.
Awesome answer. And I'm glad that our frequencies aligned and got into coherence, because I'm on board to help in any way that I can. I think part 2 will entail water, the story of how you met John, and then all the other things that we have in terms of other epigenetic perfect storms, if you will. But I agree with you, Martha. It's never a curse in that you have this ailment or affliction, because what comes out of it is better, is the world benefits, but also you've come closer and you've developed things that are helpful for John and continue to be, because I've liked the term healing or Parkinson's or life is a verb. It's not a noun, right? So Martha, I thank you so much. I'm looking forward to part 2. And I guess we could end there and set up a time for a time to reconvene.
Appreciate you having me.
Yeah.